So here we have again the two models of the muscle cell showing that they are exactly the same thing. They are just done by different artists, so they're drawn slightly differently. But both show a muscle cell with individual myofibrils inside. This one shows more myofibrils with less detail. This one shows fewer myofibrils but with much more detail. So again, one of these that you see here is one of these that you see here. So we see a bunch of them in this model. We only see a few of them in this model, but this and this are both myofibrils. Okay? So the other video that we did showed this one. We're going to work on this one now. So here we have again the individual, this whole thing is the muscle cell, or known as a muscle fiber. Each one of these, 1, 2, 3, 4, 5, 6, 7, are the myofibrils. And the myofibrils are surrounded by this beige stuff that you see here, which is your sarcoplasmic reticulum. The sarcoplasmic reticulum comes up and borders on each side these blue lines that you see here, which are your T-tubules. So if you look at the indentations that you have on the muscle cell here, on the sarcolemma, the plasma membrane, which is called the sarcolemma, you see that these are the openings to your T-tubules. If you turn it this way, you can see here that that opening that is coming right into these little blue tubes here. So these openings are simply the openings into these blue tubes, which are your T-tubules. And you see how they wrap around the myofibrils. So on either side of the T-tubule, you see that the sarcoplasmic reticulum becomes sort of concentrated here on each side, and this is called the terminal cisternae, where you have the sarcoplasmic reticulum on either side of the T-tubule. So this is terminal cisternae here, this is terminal cisternae here, and altogether the three of these make a triad. The T-tubule with the terminal cisternae on either side, that's your triad. Okay. So this is where we would store the calcium for the muscle contraction, for the sliding filament. Then over here, if we look at the individual details of a particular myofibril, you can see the dark A band here and the light, which is your I band here. Again, each one has a line at the center. So if this is your A band, this is your M line. This is your I band, this is your Z line. Then you can see here's the nucleus of the cell, and then this is your neuron coming in, your motor neuron coming into the cell. Again, you can see it's just very large. You can see things like the myelin sheath and things that you'll talk about in the nervous system. But this is that axon terminal coming in here. These are the various axon terminals spreading out around the cell. Again, they look very different than what they look like on the other model. But again, that's just a different artist's rendering, so everybody's going to see it a little differently. Your basic parts are still here. Your axon terminal coming in here. What this model does show clearly are your little synaptic vesicles. They're filled with that acetylcholine or acetylcholine, whichever way you say it. You have your little synaptic vesicles here. In your space, shown in the light blue here, is your synaptic cleft. And then the folds, again, are always going to be your motor end plate. The portion of the sarcolemma, the muscle membrane cell, the muscle cell membrane that folds on the side here, so that's your motor end plate. So again, in the neuromuscular junction, you have these three parts. You have the axon terminal, the synaptic cleft, and the motor end plate, and that makes up your neuromuscular junction. So then on the side here, we see again this connective tissue covering over the cell, so this is your endomysium. Again, you see it looks slightly different from this, but the idea is the same. It is still a membrane, has all these fibers telling you it's a connective tissue. So these on both models are just different views of an endomysium.